Okay, so this is going to be chapter 4, and we're going to talk about these things. Cell proliferation, that's dividing of cells and their growth, developing new cells. Tissue regeneration, so this is after an injury, and repair, so that's also after an injury. So to start with, in general, this concept of tissue repair, so that means that following the injury and the inflammatory response that we've talked about, then you have to sort of move to this next phase where the tissue actually has to repair itself. So there are a couple of terms here. Regeneration, well, regeneration, as the term suggests, is to regenerate or generate again. And so that's kind of a trick to remembering this. Injured cells are replaced with the same type, okay, sometimes leaving no trace of an injury. So that's regeneration. Replacement is where connective tissue may replace the original cells. So that means that if you have an injury that's extensive enough where you've broken through the basement membrane and the cells just can't figure out where they used to be in there, and you've done too extensive of damage to just regenerate those, then you have to replace those with another kind of tissue that will still protect you, even though it may not have been as pretty as it was the first time. Now, most cells or most injuries do result in some kind of replacement. So some kind of scarring usually occurs in any kind of an injury. Usually it's, if it's small enough, it's not something that you can see. So both regeneration and replacement often occur with common injuries. So here are some terms. Parenchymal tissue, the tissue is the name used to describe functional cells of organs. So that's your key functional is parenchymal. And stromal describes the supporting connective tissue, the vessels, nerve fibers, extracellular matrix, the stuff that's surrounding it. So like in the case of like a kidney or something like that, then the nephrons might be the parenchymal tissue, and then the cells or the nephrons and like the collecting ducts, and then that extracellular tissue, the fibrous material and stuff like that surrounding these things would be the stromal tissue. Okay, so usually when we talk about an organ, we talk about its functional cells. So that's usually pretty straightforward, although when you just look at it in this context, it seems like it could be just about anything. But when you actually think about what an organ does, then it kind of starts to make sense. Okay, so cell proliferation versus cell differentiation. Proliferation is going to refer to increasing numbers. So division. Differentiation is going to be when it changes to maybe a different type of cell. So we hear a lot about stem cells, and stem cells are pretty much, depending on the stem cell, they're undifferentiated or they're minimally differentiated. So the stem cell starts out and it doesn't know exactly what it wants to be, and so it will then, depending on where it is, like if you're talking about, a newly developing organism, then some of those cells will differentiate and maybe become a cardiac cell. Some might become a neuron, or some might become the blood cells or something like that. So that's what differentiation is. And then once a certain number of cells have differentiated, then proliferation can take place. And so those cells, this is a cell population. So if it's a population of hepatocytes, then you can have proliferation, and that cell can then be on its way to form a liver. Of course, you're going to have hepatocytes, which are going to be more of the parenchymal type of tissue, and then you're going to have supporting structures around it, which are going to be the stromal tissue. So there are lots of different tissues involved, but they still have to, as it grows, they still have to divide, and those particular cells have to proliferate. Okay, so there's the cell cycle. Okay, so the cell cycle just refers to the pattern whereby cells will either be dividing, or they're going to be quiescent, or they're just going to sort of sit there and do their job. So G0 is that quiescent, that's the word it's used for, quiescent or quiet phase. Okay, that's the G0 phase. So when a cell is actively dividing, then we say that it follows these patterns here. So G1, gap 1, it's functioning and growing. The S phase, the S stands for synthesis, and the synthesis that we're talking about is DNA synthesis. G2, it's also rapid cell growth, okay, because you now, at this point, your chromosomes have doubled, okay, so now this has gone from 1 to 2, and of course you have 23 of these, so you've got now more of those, or 46 of those, so you've got more of those, one from mom, 23 from mom, 23 from dad, so now you've got more, and so it's ready, and those will line up on the opposite side, and then this gap phase is where it's doing that, and it's getting ready, it's growing, it's making twice as much twice as many organelles, and getting ready so it can survive as two different cells. And then you have the M phase, which stands for mitosis. And then usually, and I think you probably learned it this way in biology class or physiology class, your M phase is broken into, the prophase, anaphase, all those things. But we're not too worried about that. All we're really interested in is the fact that it goes through this process of division. So the cell cycle goes through this process of division, and it can then slip out of that, and you can become a cell that's not dividing. So you're either a dividing cell and you're proliferating, or you're not, and you're just kind of quiet, because that's really where it comes down to the functions that we care about. And ultimately, when we talk about cancer, that's one of the things that gets kind of interrupted. So cell cycle, orderly sequence of events. During division, there are checkpoints to check for errors in DNA replication. Okay, so that's true. Now, before I come back to that, the cell cycle is tightly controlled by, here I'll change color so I'm not writing in the same color, cyclins. Okay, cyclins, and then cyclin-dependent kinases. Cyclin-dependent kinases are just the things that are listening to the cyclins molecules to do what they're supposed to do. Okay, so these guys work together. The cyclins are released, and then the cyclin-dependent kinases will then follow up and kind of do what their job is, and that is to contribute to this. So here we see cyclin E. So cyclin E causes progression from the G1 phase to the S phase, cyclin A from the S phase to the G2, and then cyclin B from the G2 to the M. So you kind of get the idea. Basically, what we've done, not me, but is named the molecules that we found that if you get rid of this, then cell progression doesn't take place, or if you block it, cell progression doesn't take place. If you turn these on and you apply those artificially, then it will progress whether it wanted to or not. So they basically just discovered this group of molecules and found out that they're involved in progressing from one to the other. Now, along the way, that's what this is talking about. Along the way, there are checkpoints that says, okay, is there DNA damage? Okay, so this is a process that happens where you have sort of a follow-up as the DNA is kind of unwinding. It's supposed to be DNA. As it's unwinding and then copying to make another one, then you sort of have this protein that's following along behind it, checking to make sure that G's go with C's and T's go with A's and that kind of thing, just to make sure that there aren't any obvious errors. Now, mistakes can happen, obviously, because we do have mutations that aren't checked, but in general, we've got this little process that sort of goes along and makes sure that there isn't anything wrong. So, to make sure that these guys haven't fused together or something like that. Okay. So we have these checks and balances and control, and that's really the thing. You've got control of the cell cycle, and then you have checks to make sure, as well as you can, that there haven't been any mistakes that are made. Okay, so who says at what point does this cell? Yes, we've learned that the cyclins are in charge of progressing through the cell cycle, but who in the first place? I shouldn't say who. What? The answer is right here. What in the first place says it's time for this cell to start proliferating or dividing? And those are growth factors. So growth factors are just another group of molecules, proteins, that increase cell size and cell division. So Here are some that are listed: platelet derived growth factor, fibroblast growth factor. So it kind of depends on which receptor the cell is expressing as to what's going to bind to it. And then once this growth factor, and a lot of times these growth factors are just kind of there in small numbers, they might increase after an injury or something like that, but these growth factors are just sort of there. Or not there. I mean, they may not be there at all because you don't want any cell division, but usually they're there in fairly small numbers for cells that are kind of trying to maintain an organ or something like that. And so the receptors are there and they're activated, and then it causes, and of course we don't have to know all of these little details, but it causes a whole series of intracellular events to take place. Okay, so all of these things are phosphorylating each other, they're activating each other, calcium is involved. So all kinds of things happen, and then you have the result, the ultimate result is gene transcription, and then you have the activation of these cyclins, and then the cell will start to divide. Okay, so then you turn on all of the genes that are in the nucleus that are involved in cell division. Okay, so that's what growth factors do. Okay, so so here is another example: plasma cell proliferation. Growth factor attaches to the receptor. This is showing what looks like a B cell, and you can see that here's a growth factor that says, hey, it's time for you to divide. Whatever you've got here is reacting, and so or we're having an immune response. Something has invaded us, so it's time to make more of you. So the growth factor comes down. You release more of it. It binds, and then these guys start to divide and proliferate. Okay. So the proliferative capacity of tissues. So we have different tissues. I just mentioned that some cells divide, some cells really don't divide. And that's really what this whole slide is about. So we have some cells that are continuously dividing, and it sort of makes sense that they would be skin cells, GI system, things that are kind of getting a lot of use, so to speak. So your skin is constantly being sloughed off. Anything that's kind of exposed to the outside, something like that, it's easier, rather than trying to maintain those outer cells, it's easier to just let those kind of fall off and then replace them or regenerate them. So we see that on those kinds of tissues. Usually some kind of epithelial tissue is where we see continuous division, and so it's constantly proliferating. And then we see kind of this in-between phase. And by in-between, I'm talking about these tissues down here that don't divide at all. So in between that are these stable tissues that stop dividing when growth ceases. So at some point, your liver or whatever, these cells have been made. So you have your hepatocytes in there. They've been made, and they're okay. They're doing their job. And so as long as they're doing their job and they aren't sending off any errant signals, then they will just sort of sit there, and those particular cells won't divide. They'll just sit there and do their liver job. But it's only when there's been some kind of damage that it turns on the so if you have a partial hepatectomy, then maybe you're going to need growth factors, and some of these cells will have to start dividing to replace the ones that have been lost. That's my bad drawing of a liver. Okay, so parts of the kidney, liver, smooth muscle. So smooth muscle is able to regenerate in certain places. Vascular endothelial cells, so that means your blood vessels, and maybe your lymph vessels, things like that. So those things, when needed, can regenerate. Okay, so they can start dividing. So we call those stable. Permanent means that they don't regenerate. So they remain in that G0 phase. So they switch over into that. You have cell division when the organism is being formed, and that goes on for a while, and then at some point it stops, and that's it. You've made all that you need, presumably. And so if these are damaged, they aren't replaced, they aren't regenerated, they aren't regenerated, instead they're replaced by non-functional scar tissue. Okay, so that includes neurons, skeletal muscle, and remember this, skeletal muscle, cardiac muscle. These are permanent tissues. Okay, so continuously dividing, stable, and of course the stable tissues, a lot of times you have some kind of apoptosis, cell death, and so you have a small amount of regeneration that's taking place to replace those cells that are dying. All right, so stem cells. It used to be thought that there weren't any stem cells in the body. That was long ago. We now know that there are some stem cells even in an adult. But basically what a stem cell is, okay, well, let me just say also that it used to be thought that stem cells were just stem cells. They were they were undifferentiated cells. Okay, they could become anything that they wanted to be. But now we know that some stem cells are slightly differentiated and they can't become anything. And there's been a lot of research on that. So a stem cell is incompletely, so that's the word we use, incompletely differentiated throughout life, which means that sometimes they can divide, and maybe you want this type of cell to come from it, maybe you want that type, maybe you want that type, or maybe you want that type. Okay, but that's it. Those are your choices. Okay, you can't become any kind of cell. In this case, you're only going to become these types of cells. And so the stem cell is that first cell up there. It says, you know, I can become anything I want, and depending on what kind of stem cell, that may only be a few things. Like for example, the multipotent stem cells in your bone marrow can be, so I don't know what I am. I am a multipotent stem cell, which means that I can either become a red blood cell, a white blood cell, or a megakaryocyte or a platelet. Okay, so those are pretty much my choices if I'm this multipotent type. So how does it do this and still maintain the stem cell? You have a different kind of division that takes place. So here's the stem cell to begin with. If we draw the original stem cell, then it will divide. Okay, one of those divisions will produce the same stem cell that was there before. Okay, so it produces the exact same stem cell. Here we'll color this blue to show that it was the same one. Then you have another stem cell that can go on to differentiate. So that way you're maintaining, hope that makes sense, you're maintaining the population of stem cells, so it's not like this guy divides and becomes differentiated, both become differentiated and your stem cell is gone. One of these will go back and will maintain its identity as a stem cell. The other is the one that goes on to differentiate and become different types of cells, potentially. Okay, and then these can divide and become something depending on the tissue. Okay, so they're capable of self-renewal, and that's what we see here. We say that here's the stem cell to begin with, division, and then you've created another one that's exactly the same. Okay, asymmetric replication, again, that's where we talk about it's replicating, it's dividing, but it's asymmetric, which means it's not dividing the same. So one of these is becoming a stem cell as it was. One of these is going to go on and be a differentiated cell. All right, so one differentiated, one remains undifferentiated. And there are different potentials or potencies, and we call these the potential to become whatever it's going to become. Totipotent means that it's the least differentiated. It can become anything, okay? So its future is wide open. Pluripotent, these are what we call the embryonic stem cells. Multiple cell lines are possible, but you can't take this and create a completely brand new organism. Okay, so you can create just about any kind of cell in the body. So from an embryonic stem cell, you might be able to create a brain cell or a liver cell or something from the kidney or any number of cells, but you can't create a new organism necessarily from that, or sperm cells or something like that. You aren't able to produce those. Multipotent, that's what we see the most. Okay, so multipotent is where and the best example that you'll see are these hematopoietic stem cells. Those are the ones that can become red blood cells, white blood cells, or platelets. So that's the important one. That's the important one to remember. And then unipotent is the most differentiated. It can really just basically become another cell like itself. So we found adult stem cells, some of them in different stages of differentiation. And that's what science is doing a lot. A lot of research is being done on this, is to take this stem cell that can become a red blood cell or various types of stomach cells. You have different types of cells in the stomach. And so they found these and they say, okay, well, it can become an interchromaffin cell or it can become a chief cell. But what if we can turn back the clock to make it, instead of being multipotent, maybe make it pluripotent. Just figure out what genes were turned on to make that difference, to turn it from a pluripotent into a multipotent, and then maybe figure out a way to turn those off and go backwards, and then maybe go backwards this way, and then maybe we can get to the point where we can actually take a cell from you and then turn back the clock on that cell and then have it produce cardiac cells, and so to replace the cardiac cells that have been lost, like if someone has a heart attack or something like that. So far, what that's done is it's created tumors. That's been a big problem. Is that we've tried to turn back the clock, and when we do so, sometimes we miss some of the controls, and we end up with a tumor being developed, and that's not good either. Okay, so there's still some work to do on that. All right, so the extracellular matrix, that's a term that I've used before and maybe one that we already understand. So here's an example. This is epithelial cells. That could be your parenchymal cells. And then you have in here the extracellular matrix. And down here you have a blood vessel that's going along here. Okay, so the extracellular matrix is basically that. It's outside the cell. Now, that doesn't mean to say that there aren't any cells present in the extracellular matrix. It's just saying that the organized, so this is where the organ is, this is where the blood vessels are, and so these are more organized types of structures. The extracellular matrix isn't as organized. It's important, and it's organized in its own way, but it's basically just filling in that space and serving as a protective area or cushioning area or something like that. It's necessary, so don't get me wrong on that. It is necessary. All right, so if we sort of look at this, we can see, so here are some of these cells. Here's a fibroblast right there. Okay, a fibroblast, what do you think it might be making? Fibroblast, it's making these fibers. So here's hyaluronic acid. There should be some collagen. So here are big collagen molecules, collagen proteins. These are all proteins, and that's going to give strength to your tissues surrounding your organs and within your organs even. And so these fibroblasts are constantly making fibers. Okay, so that's where they all come from. So that's kind of what's laying down, and that's important because we're going to hear the word fibroblast a lot. So it's worth sort of taking a picture of what it is and what it does. And that's what it does. They're scattered around here, and they're making these fibers. So they're making these fibers for the extracellular matrix. So the components of it: fibrous structural proteins, collagen, water hydrated gels, hyaluronic acid, and adhesive proteins, integrins. So that's what we see here. Okay, so those are also an important part. A lot of times they're holding on to things to kind of keep it all together, so to hold it all together. All right, so if we have damage, there is a good chance that we're going to have damage to connective tissue. Okay, so if we have that's what I want to highlight. We're talking about connective tissue and repair of that connective tissue. So when regeneration can't occur, scar tissue is generated, and that's what this process is. So angiogenesis, so one of the first things we need to do if we have a big chunk of skin taken out is we have to put new blood vessels in there. So angiogenesis is this new capillary. So new capillaries are put in growth of granulation tissue. Now granulation tissue, I understand, is a bit of a vague term because really what it means is it means a lot of extra stuff that looks kind of granulated. And so the granulation tissue consists of these new capillaries that are forming, the fibroblasts that are in there. In the case of this connective tissue, inflammatory cells, because you just had an inflammatory response, so you may have a lot of neutrophils and macrophages and things like that. And so that's the first thing that you sort of see are these leftovers and this first attempt to try to start to fix the problem. And then you kind of progress. So that's angiogenesis and ingrowth of the granulation tissue. Then you sort of progress to this, and you start to bring in more fibroblasts. The capillaries get a better foothold. You start to bring in more fibroblasts, and these fibroblasts actually start to lay down some of this extracellular matrix, so some of these fibers that we just talked about. Growth factors direct fibroblasts to enter the developing extracellular matrix. Dense collagen fibers are synthesized, okay, and that all forms a scar. So your angiogenesis and your first attempts are kind of this new stuff moving in, and it doesn't look very organized. And then you progress to this, and you're actually starting to lay down the extracellular matrix there. So maturation and reorganization. So maturation and reorganization, it's not that difficult to understand. This says collagen synthesis slows to match the rate it is degraded. Okay, well, if you have a bunch of collagen molecules that are kind of like this, and then as you're stretching your skin and you're using it, well, maybe some of these will break and die, okay? And then you end up with collagen that's sort of laid down in a more organized way, maybe the way that it needs to be organized for you. So most of them may be in this direction. Okay, so that's where you need the strength, or that particular body region needs the strength, rather than just sort of being uncoordinated and not as helpful. So many of these die, and then they're replaced, and you kind of have this reorganization that makes sense. Okay, all right. So usually this whole process begins within 24 hours. Okay, so you have this initial inflammatory response, you have the initial injury, and then this connective tissue will start to repair itself within 24 hours. How long it takes depends on how extensive the injury was. So, and it's evidenced by the migration of the fibroblasts and the induction of fibroblast and epithelial cell proliferation. So we can start to see things developing at that point. Okay, so here's some granulation tissue, and you can see that the injury is not far. It hasn't been that long since it happened. And so you kind of can see that, well, we're not really rebuilding anything, but there's kind of a mess, and it looks like the body is trying to respond in some way. Okay, so it's trying to respond in some way, so we can sort of see some of these fibroblasts. We can sort of see some of the fibers, maybe a few new capillaries that are being put down. Here we can see more of these organized strands of collagen. So we can see the collagen sort of in there, and so you get some of that reorganization that's taking place to make that collagen line up in the way that is going to be most beneficial for, in this case, the heart. All right. So cutaneous, so that was connective tissue. Now we're on to cutaneous, so skin. So when it comes to wound closure, so if you injure yourself, and it really depends on the type of injury as to how this wound is going to heal and how we are going to let it heal. Okay, so primary intention is when you have a wound, or first intention. It's also called first intention and second intention. So primary intention is when you have a wound that basically is a surgical cut or a knife, or just kind of you cut yourself with a knife in the kitchen or something like that, where you can actually pull that tissue back together. So that's good. That's ideal, because if you look, well, if you had let that stay open, then you would have a huge gap here that your body would have to fill. Okay, a lot of connective tissue would have to be dumped in there, and you would have to fill that. But as it turns out, if you can sort of pull it back together, then that really helps with the healing process. And then you just have this little area here that you need to bring in the fibroblasts, and you need to have epidermal or epithelial cell division, proliferation, and you try to rebuild it the best you can. That's also going to reduce scarring, obviously, because this skin will still be maintained here. Now this will have to be filled in with connective tissue, so you might have a little scar there, but it's obviously much better than this, which is where it is so opened up that you can't pull it back together, and that's what we call secondary intention. The wound is too extensive to bring the ends back together. So that's where you sort of have to allow the granulation process to take place. You have to allow that to be filled in, the connective tissue to be filled in, and then you can have healing. So it's going to take longer, so there's a longer repair time. Obviously, it's going to have greater scarring, better chance, I shouldn't say better, a higher chance of infection. So in this here in the primary intention, you have less of an opening, so you don't have as much chance for bacteria to get in there. It's easier to keep clean. This is much more difficult to keep clean because you don't necessarily want to clean it because you've got all those brand new capillaries that are trying to grow in there. So it's more difficult to clean without damaging what your body is trying to do to fix it. However, you do want to try to keep it clean. Because you don't want bacteria to go in. So it's really a tough situation there. More exudate, so that means that more stuff is going into this gap, moving out of the cells into this extracellular or outside the cell area. Greater need for care, obviously, and burns, ulcers, large surface wounds fall into that category. All right. So what are the phases? The phases of cutaneous wound healing. So this was, these were our intentions. So we had already talked, I'm sorry, I'm skipping around, but here we had the phases of connective tissue repair. So now we're going to talk about the phases of cutaneous wound healing. So this is on the skin. Well, number one, we can already check that off the list because we've already talked about the inflammatory phase. We know what happens during inflammation. And that, of course, is going to happen at the time of injury. Okay, you have hemostasis, which is basically a platelet plug. You're stopping the bleeding, platelet plug formation. We'll probably talk about a little more about that later. We've already talked about things like fibrin, fibrinogen turning to fibrin, and fibrinogen turning into fibrin, and then forming that platelet plug. The platelets are in there. Some blood vessels or some blood cells are in there. I have a bad habit of using the word blood vessel instead of blood cell. I've noticed that. So you sort of form this platelet plug or this plug. So that's one of the first steps, and part of the inflammatory process happens, and then this platelet plug forms. So that's kind of a bit more advanced in terms of the inflammatory response. But then the vascular and the cellular phases of inflammation. Remember, the vascular phase means that you have more fluid moving into the area. The cellular phase means that you have white blood cells that are moving into the area, and the cytokines that they're releasing to bring in more white blood cells to try to kill any kind of invader. So we've already covered all of this stuff that occurs. So the next phase is the proliferative phase. So inflammation has taken place, and now we have to, whatever happened, we have to move to the next step, and we have to start to try to fix it. And so that's about, whereas this happened, whereas the inflammatory phase happened right away. Remember, those neutrophils were on the job right away. The proliferative phase, 2 to 3 days after the injury, that's when you have building of new tissue to try to fill it in. Fibroblasts start to, remember I pointed out the little fibroblasts in the connective tissue, those little guys shaped like this. They're going to start secreting collagen and other fibers. Okay? Collagen is the strongest one, so that's the one we mostly talk about. Fibroblast secretion of collagen, angiogenesis of new capillaries. So I've already used red, but new capillaries will start to form. Okay, which is angiogenesis. Okay, so then you kind of get a blood supply into that area. Granulation tissue is laid down for epithelial cells to grow on, so that means that's all part of this, the fibroblast formation. This is all part of this laying down of the granulation tissue. You've got sort of a matrix or something from which everything else can grow. And so you need stuff in there, and these are the first guys to go in. These fibroblasts, some of the collagen, the new capillaries start to thread their way in, and so that initially is granulation tissue, and then the epithelial tissues will start to kind of form over that the best they can, and where they can't grow, it's filled in with connective tissue. And then we have this next process. It's pretty much the same process we talked about with connective tissue, the loss and gain of collagen. Okay, so it's the same deal. If you're pulling on it in this way, or up or down, or you're moving it around, then the collagen is going to have to reorganize and give you the strongest type of cell, or of healing area that you can get. Okay, and that usually happens about 3 weeks after the injury. Okay, so what does this all depend on, and how can you speed up this process, slow down this process, or how is this process limited? A lot of times it just comes down to your nutritional status. So you're going to need proteins, which provide what? They provide amino acids. So you eat proteins, which are then broken down into amino acids, which can then be built back up to make the proteins that you need. Okay, so when you're going through this healing process, you need to make sure that you have all of these things. You have enough carbohydrates for energy, you have enough fats to make new cells, because you have phospholipids around the new cells. So you need all these things for pretty clear reasons. Proteins to make other proteins. So you need the amino acids to make all these proteins that are going to be giving these signals. Carbohydrates for energy, ATP, that's going to be involved. And then fats also for energy, but mainly because you need fatty acids because you're rebuilding cells, and all those cells have fatty acids in them. Vitamins to help speed things along, and we know what a lot of these vitamins do. Vitamin C for collagen synthesis, vitamin A to make this epithelial tissue capillary formation and collagen synthesis. B vitamins they're co-vitamins or coenzymes for enzymatic reactions. Vitamin K is involved in the clotting process. So all of these guys are necessary. And then of course minerals, and that includes sodium, potassium, chloride, and this one, calcium, all of those minerals. Plus, you need magnesium, and then you've got blood, so you need iron. Okay, so you need all of those things too. Okay, well that's about it. Wound healing in the elderly. One thing to keep in mind is they have decreased dermal thickness. So you have the epidermis out here, and then you have this dermal area here that contains some of the nerves and fat tissue and things like that, blood vessels. The dermal region in the elderly is much smaller. Okay, so they lose some of their elasticity. Their skin becomes a little more fragile, and it's easier to cut through. And a lot of times when you cut through it, it can be a little more traumatic. You may find yourself with a very deep wound, which would be actually physically deep in a younger person, like in a child or something like that. You may have to cut quite a ways through. Meanwhile, in the elderly, a small knife cut would be able to actually penetrate completely through the dermal layer. Which is not as good, and a lot of times you need to get medical treatment for that. So they have a decline in collagen content. Collagen provides a lot of structural stability, and so that's one of the things that makes it a little more fragile. Loss of elasticity, so it doesn't stretch as well. So you've lost its ability to stretch, and it's a little more fragile. So that means that it's much easier in that respect to damage the skin of elderly people too. More vulnerable to chronic wounds such as pressure. Diabetic, so pressure is going to sort of choke off that area to blood flow. Diabetic people or diabetes can damage blood vessels. Ischemic ulcers as compared to so ischemia, meaning again this loss of blood flow that can result in ulcerations or open wounds as compared to younger persons. Okay, well that's it for this section.